But the other big area of science that has been advocated in the past year is that of the interrelationships between animal and human health. And of course, this, as I say, aligns with our area of work in protecting animal, plant and human health. And indeed, I'm delighted now to be moving in to our first scientific session of the day, which will focus on the interactions between animal and human health. And to chair the session, I'm delighted to have Professor Stuart Elborn with us. Professor Elborn is Pro-Vice-Chancellor of Queen's University for the Faculty of Medicine and Biological Sciences. And indeed, you know, as Colin has highlighted, we have an ever-growing and strengthening alliance with Queen's University, which we're delighted about. And Professor Elborn has been part of the team working on that agenda. But as well as that, this time last year, as we all know, all of a sudden we had to work from home very quickly because of the COVID-19 pandemic. And very quickly, Professor Elborn pulled together a consortium of AFBI, Queen's, UU and ALMAC, working with the Department of Health to respond very quickly to Northern Ireland's response on how to deal with the COVID-19 pandemic. So Professor Elborn, it's great to have you here with us today to chair this session on interactions between animal and human health. And indeed, we've very much enjoyed working with you throughout the year to deliver a lot of the testing under the COVID-19 response. So folks, just to brief you as well, what we'll do is we'll go through the presentations and then we'll have a panel discussion. And we would be very keen for you to submit your questions through what I think and hope you have a question and answer icon on your YouTube screen there. So please do submit your questions through that Q&A function, and we will take those questions to the panel session when the presenters get through their presentations. So Stuart, great to have you with us. Thanks so much. And over to you now to chair the session.
Thank you very much, Elizabeth, and thank you for the very kind invitation to join this conference and be part of it. I also really enjoyed Minister Putt's opening remarks that really emphasised, I think, something that we've learned, particularly over the last year, that science can have real impact on what we do in a range of sectors. One of the learnings from the pandemic that I think we have really gained is the importance of working across sectors of academic disciplines, of government departments, of countries, jurisdictions. This has been a world challenge for us and really emphasised the importance that we appreciate that we're living in a planetary ecosystem where there are enormous interdependencies. I think the clear narrative, I think, that the coronavirus that is causing the COVID-19 pandemic is from an animal source, almost certainly from bats, has really strengthened and emphasised the importance of understanding the relationships between the ecosystems that we live in, and particularly the interaction between human and animal health. As Elizabeth has said, we've been able to work with AFBI and the team involved in viral diagnostics to support the diagnostics for COVID-19 and for the coronavirus. And that's been a wonderful example, I think, of intersectoral working. And we're also now working closely together around wastewater surveillance with JBC and the other 4 nations in the UK and all Ireland, so that we can have the right infrastructure for the future to protect the infection issues that are part of the human and animal ecosystems, in particularly focusing initially on coronavirus, but using what we learn from that and the infrastructure that we can build for future surveillance and health protection. And I think this is going to be really important, as Minister Putt's emphasised, in the challenges ahead. There may be further infection challenges, there may be further pandemics, but the ability to respond to the challenges of climate change and the important adjustments we need to make to our whole ecosystem to respond to those challenges are going to require this intersectoral working, which I think we've gained a lot from over the last number of months. So this session is really going to focus on the interactions between human and animal health, and we have three excellent speakers who are going to share some, I think, really important data, some policy issues, and hopefully get you thinking about this particular area that we can then pick up with our expert panel and the speakers in the discussion at the end. So our first speaker is Dr. David Mccleary. David has a background in science, communication and public administration and is currently head of the bacteriology branch in AFBI. He's been leading an integrated program of research and development and diagnostics in major bacterial pathogens causing animal infection and that have an impact on public health. He has had a career which has worked across, again, different sectors. He's been involved in science, communication and policy as part of Horizon 2020, where he led for Northern Ireland on agri-food issues, but has also worked in industrial sectors and now is, as I've said, part of AFBI. He also holds an associate professorship with the University of Agricultural and Veterinary Sciences in Timisoara in Romania. So David's going to kick us off with a talk on how science supports the agri-food industry and the relevance of that in terms of both animal health and protecting human health. So it's a pleasure and thank you, David, for coming to the conference, and I'm going to hand over to you now online.
Thank you, Stuart, and thank you very much for that introduction. And I'm delighted to be able to be part of this event today. So, ladies and gentlemen and Minister, today I'd like to discuss how science supports agri-food industry needs and protects animal health. We know that zoonotic diseases are those which can spread between animals and humans, and approximately 60% of human pathogens are also present in domestic animals and wildlife. And indeed, 75% of the emerging pathogens are of animal origin. The World Bank has estimated that about 1 billion cases worldwide are caused by zoonotic diseases in the human population, which results in about 1 million deaths. And of course, livestock diseases don't just affect the health of the animals, they also have an impact on their efficiency, reducing it by up to 20%. An animal health problem is a production problem with an economic cost that we're estimating. The One Health concept has been discussed for some time, and it really deals with the fact that humans, animals, and ecosystems are all interconnected, as already been alluded to in the introduction. This requires a coordinated and collaborative approach across disciplines, and really can be captured in the phrase people, pathogens, and planet. And here we see just some of the interactions between the supply chain, the environment, and the end users and consumers. AFBI is very well placed to play a central role in this, given the broad-reaching and unique remit of AFBI. Of course, zoonotic diseases are not static, they're dynamic, and this can really be summarized by the pathogen, the host, the people, the animals, and the environment itself. And some of the factors that impact on the host are things like the immunostatus of the individual. We know that large populations of large sections of the population of humans are immunocompromised, which can impact on their susceptibility to various infectious agents. There are changes in eating habits, where we're eating raw or lightly cooked foods, Of course, logistics are always improving, and foods can be transported in a fraction of the time they would have 20 or 30 years ago or more. In terms of the environment, there are a number of changes happening, transformation of agricultural practices, and maybe challenges in biosecurity. The change in land use that urbanization is actually causing. And of course, the environment itself and climate change are promoting the spread of new vectors or changing patterns in vectors around different regions. And pathogens themselves are not static. They will continue to evolve according to the production system that they find themselves in. And indeed, we have emerging infectious diseases from wildlife, which my colleague Ken will mention later on in his talk. In the main, I suppose, viruses present their greatest risk to the human population because of the potential for respiratory spread and person-to-person transmission. In terms of the food supply chain of foods, bacteria are significant. And even some of the pathogens that we would consider to be very common today are very well recognized. Only a relatively small length of time ago, they weren't completely linked with human illness. Campylobacter jejuni was only really linked with human diarrhea in the 1970s. E. coli 0157 H7 was only identified as causing human illness in 1982, and that whole group of STEC is a very important one. And then if we look forward to more recent years, we can see how strange combinations or atypical forms of these pathogens can actually appear. We had atypical E. coli 0104 H4, which appeared in 2011 in Germany and resulted in almost 4,000 cases worldwide and unfortunately 53 deaths. So what can we say about the future? Well, it's unknown, but we see that there is increasing recognition of the causative agents for infectious disease. So what can we do to prevent and control animal disease and the potential for transmission to humans? Well, prevention is, of course, better than cure. And if we have a look at this range of different strategies here from left to right, we really see the ones on the left are the ones that will help to identify and hopefully control or prevent infection. And then as we move through to other strategies, we're actually trying to control or have more advanced approaches. So surveillance is critical. Both passive and active surveillance. So we can identify if there is an incursion and deal with it. Overall, just general promotion of animal health is essential, which hopefully should eliminate the need for treatments or therapeutics. And enhanced diagnostics are critical within this as well. And then, of course, if these measures are not adequate, we need to think about other aspects such as vaccine development or breeding for disease resistance. And of course, a very big issue is responsible use of antibiotics or the quest for alternatives. All of this comes under the One Health approach and relies upon the application of new technologies. And I'll give some examples. So one might ask, well, what are the impacts of animal health and welfare? Well, one clear one is that these are prerequisites for trade. We have to test food-producing animals to comply with statutory requirements. And the work that we do in AFBI actually helps to protect a market outside of Northern Ireland of 3.9 billion per year. Each year, we have a number of trade delegations auditing AFBI to hopefully demonstrate the strong systems that we have in place. And that demonstrates the high animal health status that we enjoy here and provides market confidence through the trusted supply chains. A recent example of how AFBI has supported new market access is the work that we have done in developing testing programs for the Canadian and the US markets for STEC, for E. coli 0157. And this has actually allowed the UK to export to these markets. And of course, in a post-Brexit era, access to markets is going to become even more critical. And the science that AFBI can provide will help to access these markets and secure them for the future. I mentioned technologies, and I suppose in terms of microbiology, sequencing, whole genome sequencing is a game-changing technology. It allows high density and high throughput genotyping of animals and pathogens at the highest levels of resolution. It's extremely sensitive and specific, and it's transforming how we are doing microbiology and genetics, streamlining work streams so we really have, once we get to the sequencing part, common work streams. And output data from this are digital, they're portable, they can be shared worldwide, compared with international databases, and interrogated at a later time points as we maybe recognize new variants properties. But the actual sequencing is just one part of the whole system here that's needed for this. IT infrastructure is essential, and bioinformatics expertise is really a critical resource, which is one that we need to make sure that we develop further for the future to meet the needs. So what is the outlook on these disruptive technologies? Well, if we look at a statutory type context, the same need for standardization and accreditation applies to sequencing as it does to the more traditional forms of microbiology upon which a lot of statutory work is currently based. But the benefits of sequencing are great because they can allow enhanced surveillance, genomic surveillance potentially, an outbreak investigation at the highest resolution. Very often the cause of agents are not always identified. So if we can look at the highest level of resolution, we have a much greater ability to have the resolution needed to identify the cause of agents. And of course, sequencing doesn't just identify isolates, it can actually characterize them, determine susceptibility to antibiotics, mutations, and variance properties, all in the same process. And of course, organizations such as AFBI have the capacity to conduct research on the data arising from statutory analysis and sequencing being applied to that. For example, comparative genomics, where we can actually look at the pathogen genomics and the variants associated with more severe forms of disease to see if there are certain patterns, and maybe a more targeted approach could be applied. The whole quest into host and pathogen interactions, changes in the host or differences in the host and the pathogen can have massive impacts on the outcome of disease. And then finally, looking at the very fine scale, some of the phylogenetic approaches that we have used in other contexts can be used to actually get a very clear idea about certain systems. I suppose it's fair to say that if we hadn't had COVID-19 over the past year, antimicrobial resistance would have been much more in the prominence. It's a global challenge. And one of the stark warnings has been mentioned in the Northern Ireland report, Change the Culture, but also by a UN group, is that by the year 2050, we can expect, at the most extreme interpretation, 10 million deaths per year worldwide from drug-resistant infections. So this is something where we need global action. In fact, that is happening. But what are the strategies to address AMR? Well, we can lower the burden of infection in both animals and humans, just improve their general health, so we have less need for therapeutic treatments. We can minimize the spread of AMR through the environment. We can improve the safety of water, food, and sanitation, optimize the use of antimicrobials, increase the laboratory capacity and surveillance so we can be very clear about what's emerging and be able to deal with it. And then finally, that whole raft of research that's needed in therapeutics, alternatives, diagnostics, and vaccines. AFBI is working across all of these areas. And today, I'd like to draw two examples to your attention. That is how we are, through research, lowering the burden of infections, but also our quest for alternatives. So this is a program of research within the food safety, microbiology food safety area, very much looking at intervention strategies, looking at both the host and the pathogen, understanding the host microbiome, looking at novel sources of antimicrobials, and then how these can be applied to potentially reduce the carriage of pathogens or their variants, and very much looking at approaches which are using in vitro, ex vivo, and in vivo approaches. The examples that I'll be drawing upon focus very much on Campylobacter. We know Campylobacter is a leading cause of bacterial food poisoning. In the most recent EFSA One Health report, it showed 220,000 cases across Europe in the year 2019, which was more than twice the next most common, which was Salmonella. And of course, Campylobacter normally gives a diarrheal disease, or illness, which is self-resolving, but it can go on to cause neuromuscular complications such as Guillain-Barré syndrome. So the photograph there of President Roosevelt, famously he was known to have suffered from polio, which was from an illness in the early 1920s. But that diagnosis did not consider Guillain-Barré syndrome. And looking back, the symptoms are very much aligned with that. So it could well be that he had actually not suffered from polio, but from Guillain-Barré syndrome. Campylobacter colonizes chickens, we know, and can be present in other species, but it colonizes the chicken Zika at high levels of about 10 to the 9. And EFSA have modeled that if we reduce down the concentration of Campylobacter by a factor of 1,000, we can actually reduce humanness by more than 50%. So limiting exposure is one of the critical strategies for Campylobacter. But of course, it presents both a challenge and an opportunity to industry and to the whole supply chain. And AFBI, working in partnership with 2 additional companies, Moy Park and Aranta, together with the London School of Hygiene and Tropical Medicine, Queen's University, Universities of Glasgow and Bucharest, and King Michael I of Timisoara universities, have developed a partnership approach to this issue. And through this, we have 2 industry-funded PhDs, which have been central to the work, which has very much resulted in translational research to be conducted and innovation to be applied. This has resulted in a number of high-impact publications. One of them looked at the impact of plant extracts and organic acids on the in vitro characteristics of Campylobacter. And this showed that the virulence was reduced in vitro, and that this could be brought forward then into an in vivo trial. And this showed that the body weight of the chickens was improved by the use of this feed supplement. There's improved feed conversion rate and a lower feed intake. But overall, the carriage of Campylobacter was reduced down by between 100,000 and 1 million cells per gram in the Zika, very much playing a major role in potentially limiting exposure through the supply chain. Other work then looked at it's the biggest chicken microbiome analysis that's ever been conducted. So over the 35-day production cycle of poultry, the microbiome was analysed, and what it demonstrated was that Campylobacter did indeed appear in around day 16, as we might expect. But there are differences according to production regime, and the work went on further to actually ask the question, is there a correlation between production and pathogen reduction? And this was shown that 3 different production systems, normal, high welfare, and high welfare with omega-3 supplementation of the feed, did indeed have an impact on the microbial diversity. And Campylobacter presence was linked with increased microbial diversity and other environmental drivers. And this work will go on to develop parameters to influence the chicken microbiome, to promote production factors, but also to exclude certain genera. So in conclusion then, what does this work actually say to us? Well, we know animal health, including wildlife, human health and environment are inextricably linked. We've demonstrated that, hopefully, from what we've covered. The increased rate of discoveries of drug agents is set to continue, particularly in the genomics age, where we can have untargeted approaches, where we can hopefully quickly identify infectious agents and the roots of infection. The research that I outlined into the identification of antimicrobial molecules targeted towards virulence and the role of gut health are providing solutions to the global challenge of AMR, and this work continues. And finally, a tripartite approach between AFBI, industry and academia is delivering translational research and innovation, is providing new opportunities for industry and providing a role in protecting human health. Thank you very much.
Thank you very much indeed, David, for an excellent lecture and some really compelling examples of how bringing really sound bacteriology and science related to infection, nutrition, etc., can really have an impact on not just animal production, but have real reach through into improving human health as well. So thank you very much, and we look forward to bringing you back for the panel. While we're just waiting for the next speaker to join us, I just thought I'd pick up a couple of comments from David's talk. He ended up with, I think, some really interesting comments on diversity. We're really increasingly understanding in human health that gut microbiome diversity is important for health, and in my own area of lung infection, where a number of diseases are associated with chronic infection, that one of the ways that we see improved health parallel is with increased diversity in the airway microbiome, for example. So this is a very common theme, I think, across human and animal health, that diversity is a good thing. So it's now a pleasure for me to introduce our next speaker, Dr. Ken Lemon. Ken and myself and Ken's boss, Alistair, have had a number of very positive and constructive interactions over the last year, as Ken has been really key to the delivery of the PCR testing for the coronavirus causing COVID-19 in AFBI. Ken's an alumni of Queen's University, where he undertook his PhD in areas related to vaccines. He then went on to do some postdoctoral studies, particularly focusing on respiratory syncytial viruses, and then spent some time working in AstraZeneca, getting what I think is really important industry experience. I think it's so good to know that many of those who are driving the science in our ecosystem have worked across universities, government bodies, and industry. He has done some important work on influenza virus with AstraZeneca, and then in 2016 he joined the virology branch of AFBI as a principal scientific officer, with particular responsibility for epizootic testing and contingency planning for emerging viral diseases. And we were so glad that he had that experience in the animal sector and was able to bring it into human disease during the pandemic. Ken is going to talk to us about an area which is of absolutely key importance at the moment, and will continue to be, because it's not just coronaviruses, but it's influenza viruses and potentially other viruses that transition across the human and animal interface, and not only present health challenges in the food and agriculture sector, but translates into human health. So it's a pleasure, and thank you, Ken, for joining us to talk to us on the topic of viruses at the human-animal interface. Over to you, Ken.
Thank you, Stuart, for the introduction. Okay, so we've just heard from David about the impact of animal health on human health in terms of emerging bacterial pathogens. But in my presentation, I want to discuss emerging viral threats at the human-animal interface. And I'm going to take a particular emphasis on influenza viruses. So recent decades have witnessed a real acceleration in the rate at which new infectious diseases have emerged. Multiple factors have contributed to this, and David covered many of these, with things such as habitat destruction, human encroachment on wildlife, intensification of livestock farming, and the long-range transport of people and animals have all contributed to this. And it's really created ideal conditions which favour the emergence of new diseases. And the interfaces between humans and animals are critical to this process because this is where cross-species infection occurs. Numerous threats have already been identified as shown here, but most threats are actually unknown. And it's been estimated that there are approximately 1.6 million unknown viruses in mammalian and avian populations. And of these, up to 700,000 have the potential to cause disease in humans and livestock. And of these, only 0.1% have been identified to date. The current coronavirus pandemic that we're all living through is really a textbook example of an emerging zoonotic virus at a human-animal interface. Here we have a previously unknown virus originating in bats, transmitted to humans, probably via an intermediate mammalian host. And the human-animal interface in this example was a busy seafood and live animal market in the city of Wuhan in China, where exotic wildlife and domestic animals can be purchased for human consumption and slaughtered right in front of the customers. So we know following the emergence of SARS-CoV-2, which is the virus which causes COVID-19, the virus rapidly spread around the world, causing massive societal disruption that we're all living through to this day. There have been over 120 million confirmed infections, over 2.6 million deaths, and not only infections and deaths, but there's been a huge economic cost with the loss of 400 million jobs and an estimated cost of $28 trillion by the end of the pandemic. So although this pandemic seems unprecedented, pandemics have actually occurred regularly throughout human history. And even in the past 100 years, there have been 4 pandemics caused by influenza virus alone. The most famous of these, the Spanish influenza pandemic, occurred in 1918, and it infected up to 1/3 of the global population and killed at least 50 million. Subsequent pandemics in 1957 and 1968 killed around 3 million people, and the swine flu pandemic of 2009 killed 360,000. So in every case, the virus responsible originated in animals. The first 3 pandemics were caused either directly by an avian strain jumping into humans or following a reassortment event between an avian and a human strain. The 2009 pandemic was obviously caused by a swine strain crossing into the human population. And in each case, the viruses were totally novel. They'd never been seen before by humans, and so we had no pre-existing immunity, and therefore they wreaked significant havoc. To understand the threat caused by influenza viruses, we need to understand their ecology in terms of the various animal reservoirs in which they occur. Influenza strains are classified by 2 proteins that occur on their surface called HA and NA. There are 16 different HA and 9 different NA that make up the various subtypes occurring in wild birds. So for example, H1N1, H5N8, etc. Most subtypes are readily transmitted from wild birds to domestic poultry. And from poultry, certain subtypes can be transmitted to humans, either directly or via pigs. So in this presentation, I'll focus on influenza in birds and pigs and the threat caused by each to both animal and human health. I'll also discuss a newly discovered influenza virus occurring in cattle that may have the potential to infect humans also. So I briefly want to mention the various influenza testing activities carried out by AFBI. Surveillance is carried out using a number of accredited tests, either molecular testing looking for the presence of virus or serology testing looking for the presence of antibodies indicating previous exposure to virus. Surveillance can be broadly categorized as either passive or active. And passive surveillance includes testing of routine submissions to our postmortem department or of wild birds reported dead by the general public and tested as part of the wild bird survey. Active surveillance includes annual serological surveys of poultry or part of an enhanced surveillance following disease outbreaks. So avian influenza and the highly pathogenic avian influenza has caused sporadic disease outbreaks in poultry for a very long time. Cases have been documented as far back as the 1800s, when it was known as foul plague. And what we believe happens is that low pathogenic strains of the H5 or H7 subtypes are occasionally transmitted to poultry. And once in poultry, they may begin to mutate and develop highly pathogenic characteristics that are associated with high mortality. In the late 1990s, a new strain of virus evolved in the live animal markets of China. And this new strain of H5N1 had two unusual characteristics. Firstly, it was able to transmit from poultry to humans, and so far there have been over 860 cases with a 53% fatality rate. So far, there's been no efficient human-to-human transmission, but we know that this is possible. And research carried out in the Netherlands and Japan show that the virus is only 4 or 5 mutations away from gaining this ability. The second unusual characteristic about this new lineage of H5N1 was that unlike previous versions of highly pathogenic avian influenza that evolved in poultry, this virus was transmitted back into wild birds. And once it entered the wild bird population, it rapidly spread around the globe by birds undergoing their annual migration, and is now present in over 80 countries. During this time, the virus has continued to evolve and is reassorted with other local strains of avian influenza to produce other variants, such as H5N8 and H5N6. In Northern Ireland, we knew, or we expected, that migratory waterfowl would eventually bring the virus to our shores. During this time, the wild bird survey became increasingly important as it provided an early warning system to alert us to the presence of the virus. And indeed, the first detection of high-path avian influenza in Northern Ireland occurred in January 2017, when a wild swan was found up at Loch Beg and was submitted as part of the wild bird survey. Testing at AFBI identified the virus as highly pathogenic H5N8. Since then, numerous detections have occurred most years in Northern Ireland in a range of bird species, primarily swans, but also in geese and raptors. And various subtypes have been detected by AFBI, including H5N8, H5N6, and H5N3. So we've known that the virus is present in Northern Ireland since 2017, and our major concern was that the virus would spread into domestic poultry. Now, you know that the poultry industry is obviously very important to the Northern Irish economy, as the poultry industry is the largest private employer and has a huge annual turnover. And international regulations specify actions that must be taken whenever high-path avian influenza is confirmed in poultry. These include depopulation, restrictions on movement, and the creation of 3-kilometre protection zones and 10-kilometre surveillance zones around the affected premises. Confirmed high-path avian influenza in poultry can be highly disruptive to normal business and can have implications for international trade. On the 1st of January this year, AFBI received reports of a suspect flock of 16,000 birds that were showing nervous signs. Samples were submitted and rapidly determined as high-path H5N8. 4 days later, a second premises of 31,000 layers also tested positive. Protection zones and surveillance zones were established and the sites fully depopulated. During this time, there were numerous suspect premises, but these were all negated by AFBI, and thankfully the outbreak was contained to these 2 premises. So although we know that H5N1 can infect humans, until very recently, the risk from H5N8 was considered very low. This has recently been revised due to an outbreak of high-path H5N8 in 7 poultry plant workers in the Astrakhan region of Russia. So although disease symptoms in this case were mild, this again highlights the threat posed by influenza at the human-animal interface, why vigilance is required. So I next want to consider influenza virus in pigs. Swine influenza is one of the most important production diseases in pigs and is associated with substantial economic losses. It presents in 2 forms, either as acute epidemics of respiratory disease or as a contributor of more chronic conditions such as porcine respiratory disease complex. SIV also predisposes pigs to secondary bacterial infections leading to increased antibiotic usage and has implications for AMR. Pigs are also considered to be important in the emergence of new human influenza strains with pandemic potential. Pigs are thought to be mixing vessels as they're readily infected with both human and avian strains. And during co-infection in pigs, 2 or more viral strains can reassort, producing novel variants that infect humans. Reassortion of avian and human strains was responsible for the 1957 and 1968 pandemics, and we know that a swine strain emerged from central Mexico in 2009, causing the swine flu pandemic. Evolution of influenza strains in pigs continues to this day and continues to threaten human health. And recently, a new strain of SIV called genotype 4 has emerged in Chinese swine herds that is capable of transmitting to humans and considered to have significant pandemic potential. So again, this underlines the importance of monitoring viruses at the human-animal interface. At AFBI, we're actively involved in SIV research and development. Unlike avian influenza, surveillance of influenza in pigs has occurred less frequently in Northern Ireland, and it's currently unclear what subtypes are in circulation and what their prevalence is. To address this, we've recently initiated a major SIV surveillance project in collaboration with JMW Farms. As part of this project, we hope to not only characterize which strains are currently circulating, but also better understand the transmission dynamics on farm across different production age groups to enable more effective targeted interventions. We're also involved in the development of new SIV vaccines. We've developed an experimental SIV infection model in pigs and established assays to measure the immune response to SIV infection and vaccination. And this model has recently been utilized to test new SIV vaccines for commercial customers, but we're also planning to use it to test our own vaccines developed in-house. So the final type of influenza virus I want to discuss is a recently identified species that causes respiratory disease in cattle. Bovine respiratory disease is a major issue and responsible for around 1/4 of all cattle mortalities in Ireland. This is a major research area in AFBI, and we have ongoing collaborations with colleagues in the Republic of Ireland and America as part of the US-Ireland R&D partnership, looking at various aspects of the disease. BRD is a complex disease and involves the interplay between pathogens, the host, and environmental factors. But viruses are thought to be important initiators of disease. And historically, viruses such as RSV and bovine herpes virus were considered to be the major viral initiators. However, in 2011, a new type of influenza called Influenza D virus was discovered in US cattle. And there's now overwhelming evidence that the virus causes or is a major cause of bovine respiratory disease. The zoonotic potential of Influenza D virus is currently under investigation. The virus has a very broad host range, and not only does it infect cattle, but it can infect pigs and small ruminants also. And we know that workers with occupational exposure to cattle have shown to have antibodies to IDV. And the virus has also been shown to be capable of infecting the respiratory cells which line human airways. AFBI was the first to confirm that IDV was circulating in the UK. So during the winter of 2017 to 2018, we tested all fallen stock submitted to AFBI with confirmed bovine respiratory disease and found IDV was present in around 9% of cases. So because of this, we're currently developing, there's no vaccine available for IDV, and we're currently developing one to help to reduce the disease burden of the virus. So in the last few minutes, I just want to quickly finish off by looking at the future in terms of what we can do to manage the current pandemic, and also how we can prepare or possibly prevent the next one. In terms of the current pandemic, massive expansion of testing has been critical in the effort to turn the tide. AFBI's experienced in dealing with major outbreaks of disease in animals that require large volume, high throughput testing, such as during the foot and mouth disease outbreak in 2001. And again, these skills were brought into play again last year when we joined the COVID testing consortium alongside QUB, UU, and ALMAC. Here we utilised our existing high throughput capabilities they established a SARS-CoV-2 diagnostics facility, and this went live in May 2020 with a capacity for up to 1,000 samples per day. And this infrastructure provides critical surge capacity to the regional virus lab at the Royal Victoria Hospital, but can also be rapidly repurposed for any future threats. So what about the next pandemic? Can we do better? So historically, the response to zoonotic viruses has been reactive rather than proactive, and this has been characterized by cycles of neglect and then panic. However, even before the current pandemic, things were starting to change, and organizations were beginning to coalesce around the idea that we weren't helpless in the face of these threats. One example of this is the Global Virome Project. So I mentioned at the beginning of my talk that there are around 1.6 million unknown viruses out there in animal reservoirs with the potential, and some of these have the potential to cause disease in humans and our livestock. So the Global Virome Consortium is an international consortium which aims to identify greater than 99% of all such threats within the next 10 years before they spill over into humans and our livestock. With this information, they aim to characterize the geographic scope and host range of viruses, monitor the movement of viruses across hosts and regions, and identify transmission and pathogenicity markers for high-risk viruses. And this has all been made possible by advances in technology, such as next-generation sequencing and metagenomics. And again, this is an area that AFBI is actively investing in. So once we've identified the threats that are out there in the animal reservoirs, can we take any steps to mitigate the risks that are posed? So perhaps the greatest success story of the current pandemic is the speed at which scientists were able to develop highly effective vaccines to a previously unknown virus. For COVID, this took just over 300 days. However, every day that we can shave off this will save thousands of lives next time. The Coalition for Epidemic Preparedness Innovations is a global partnership launched in 2017 to develop vaccines to stop future pandemics. Their goal is to compress vaccine development times to under 100 days. And how they plan to do this, or how do they plan to do this? So investment in new vaccine platforms that can be rapidly adapted to unknown disease X pathogens is critical. The Oxford AstraZeneca vaccine is a good example of such a platform. And the reason it could be developed so quickly is because it spent years in development for other diseases, such as MERS. New technologies we've seen, such as mRNA vaccines, can also be rapidly retargeted to new viruses. Things such as universal vaccines hold great promise. So a universal vaccine towards flu, which may protect against all subtypes of influenza, are currently in clinical trials. And scientists have also begun to consider if a universal coronavirus vaccine is possible. The development of vaccine libraries for each of the 25 viral families that are known
Thank you, Ken, in your virtual absence. I think Ken's internet has frozen, although I now understand it's apparently these are glitches in servers rather than necessarily people's broadbands that sometimes freeze individuals during a talk. But thank you. That is a really both interesting and challenging talk, really reminding us, I think, of the importance of really understanding the global ecosystem in which we live, and that the countries really do need to work together across animal and human health to really be in a position to deal with the threats that are coming from zoonotic infections that are causing not just the current pandemic, but have the history with influenza of previous pandemics, and undoubtedly will cause further pandemics in the future, and other viruses that may well also come into play. I am reminded that I'm old enough to have lived through a number of flu pandemics in the NHS that weren't at the scale that we're currently experiencing, but did cause significant disruption to healthcare delivery and resulted in a high number of deaths during the particularly large pandemics. And we kind of have got used to flu and almost acclimatized ourselves to it, but it remains, I think, a major challenge for human health moving forward. And the H5N8 story, which Ken alluded to, I think is one that we need to understand better so that we can be prepared for future variants that may cause both avian, porcine and human disease. So it's now a pleasure to move on to our third speaker. It's a pleasure to welcome you, Miles, to this conference. Miles Carroll is Deputy Director and Head of Research and Development at the Institute National Infection Service for Public Health England. Miles has a long and very prestigious history of working with a number of infectious diseases in a number of contexts. He was previously head of National Infection Service Research at Public Health England in Porton, and is involved in current research investigating the importance of natural acquired immunity and host response to infection, and also how we use that understanding of natural immunity to ensure that we've got good in vivo models where we can test vaccination. This has clearly been of critical importance over the last 14 months during the current pandemic, but I think has really emphasized the importance for the future of having the right approach to vaccine development, which means we need to understand even more clearly the natural immune responses to infections, whether they're viral, bacterial, and I think an increasing risk from fungal infections. So Miles is going to talk on emerging disease threats, natural acquired and vaccine-induced immunity. And again, a pleasure to have you at this conference, Miles, and I'm going to hand over to you. Thank you very much for joining us.
Thank you very much indeed for the kind invitation and the introduction. So I'm not actually going to talk so much about the vaccine work because I wanted to concentrate on pathogens that are not just flu and SARS-CoV-2, because I think we get into a full sense of security and think that if we're protected or have a plan against pandemic flu, then that takes care of everything. I think we need to fully understand the huge array of different pathogens out there and have a preparation for each of their classes and how they transmit from animals to humans and then human to human. So we've had two fantastic presentations from David and Ken, and they've set the scene. I'm going to talk again about a number of pathogens that you may not have heard of, a bit more exotic, and a few that most of them are overseas, but a few of them that are in the UK that we didn't know were there. So this highlights the extent of the richness, really, of those viruses that are out in the environment, and it really highlights that we know very little about what's out there. I mean, Ken talked about 1.6 million viruses. I wouldn't be surprised if there's even more of that. We know a fraction of what's out there. And what this map shows is some key outbreaks that Ken touched on a little bit as well around influenza, MERS, obviously SARS-1, and it also talks about Ebola, chikungunya, and then Chromium, Congo hemorrhagic fever. The list goes on and on. And these are the ones that we know are out there because it's been linked to a number of clinical cases and deaths, and we've got molecular diagnostics to these locations or taken samples back to our labs and shown these pathogens were the cause of those infections and fatalities. But I would say there's probably this many outbreaks happening every year, not just those that happened the last sort of 10 or 20 years. And there's something common about the viruses that, that spill over, and that usually that they are RNA viruses, so they can mutate maybe 10 times quicker than DNA viruses and can adapt to new hosts. And as Ken and Dave already mentioned, there's so many of these pathogens have an animal host. I think Ken said 75%. Last time I read it was 70, but it's probably increasing year on year when we understand more about those viruses that are lurking in the animal kingdom. So at Public Health England, we have a specialist laboratory called the Rare Imported Pathogens Laboratory, RIPPLE, and is specifically dedicated to diagnosing those unusual pathogens that we thankfully rarely see in the UK. And we know that because of increased travel, increased destruction of natural environment, movement of livestock, movement of people's pets, obviously like to take on holiday with them, we get increased risk of introduction of exotic pathogens and high consequence pathogens into the UK. So we're continuously studying with international partners these new viruses as they appear, and we're developing new diagnostics all the time to ensure that the UK is able to diagnose rapidly those pathogens as they come into our country. And this is obviously done in partnership with people around the world, but and also ECDC and CDC in the US, but specifically with the WHO, who are a tremendous entity for bringing international collaboration together. So this is a quick story I'd like to bring you up to speed with, and it's all about tick-borne encephalitis virus that you probably think doesn't happen in the UK. And we didn't think that until about 24 months ago, when we had a PhD student as part of a health protection research unit collaboration with Liverpool University and Oxford and Liverpool School of Tropical Medicine. And what her role was to do some sampling, blood sampling of the deer populations in different locations throughout the UK, do some serology studies to see if there's any evidence of things like Laupinil, that obviously is an animal pathogen, and a close relative, this virus tick-borne encephalitis. And what she showed was that there did appear to be a number of hits serologically. We couldn't find any virus initially in the deer populations. So the approach was to look at the tick populations in two forests, one in Thetford in Norfolk and the New Forest down in Hampshire. And both tick populations showed PCR positive tick-borne encephalitis, and the power of sequencing, as Dave and Ken have already highlighted, allowed us to rapidly work out that this virus was two separate introductions, likely from different regions of Europe. And the most obvious way that the virus got into the UK was those ticks hitching a ride on migratory birds that do pass over those forest regions. So this is a brilliant example of pathogens in the UK that were obviously causing probable spillover events. And since that time, we've identified a couple of human cases where ticks have infected them with tick-borne encephalitis virus and been able to diagnose them. Again, when you know what you're looking for, it's all very easy, but most of the time we are working in the dark to some extent. So just switching to a pathogen again, you think won't be anywhere near the UK, a virus called Chromium Congo hemorrhagic fever, a Mania virus, identified in the 50s by British, Russian and American scientists. It's a case fatality rate of anywhere between 10 to 40%. We classify it as a viral hemorrhagic fever because obviously the associated hemorrhaging in the pathology of the virus in humans. So the virus, again, uses ticks to replicate in, and it infects a lot of wild animals. But we do know that the virus is also able to cause human diseases with unsafe slaughtering practices that do occur around the world. The incidence of CCHF is increasing in Europe, in places like Bulgaria and Turkey and Romania. And that season of infections is associated with the season where the ticks are most active and likely to infect walkers, farmers, etc. And this is the Halima tick is restricted by temperature in the region shown in this map. And you think, well, the UK is okay, we're not in there, but this tick has been located in southern parts of England. And we've had a number of cases in Spain in recent times where walkers in areas around Madrid have become infected by ticks, and there have been a number of fatalities and human-to-human spread as well. And those ticks in Spain, looks like they took their ride up from migratory birds in North Africa, because again, sequencing can help us with phylogenetics identify the origins of these infections. So I want to spend a little bit of time on Ebola. It's something I spent the last 6 years working on quite a bit, and spent a lot of time in West Africa as well. So we obviously know the big outbreak in 2013 and ran to 2016. Again, the virus has the ability to replicate quite effectively, we believe, in bats without causing significant disease in those bats. There's an interaction with bats and forest animals, including primates as well. And the theory is that bushmeat hunters will go into the forest, hunt these animals, and some of them are infected, bring them back to the village, in that preparation process become infected, and then the virus spreads on. And this is obviously what happened in the West Africa outbreak, where a young boy was thought to be playing with an infected bat in the village of Miliandu, and the virus spread throughout that village and then over to the border between Liberia and Sierra Leone and into the capitals of those cities, including Guinea. And then even if you do survive, especially if you're a male, the virus appears to be able to lay dormant in specific, what we call immune privileged sites, like the eye, the central nervous system, and the testes. So PHE Porton Down are a member of what's called the European Mobile Laboratory. It's an EU-funded program from mid-2013. And it was a system where you would respond, you would prepare for an outbreak, primarily a human pathogen, but it could be animal if that's where the governments want us to go to deal with. And you train in a tent, basically, in a military base in Germany, as if you're dealing with a category 4 pathogen like Ebola, and you apply different diagnostic approaches. So in March 2014, there were lots of reports from ProMed that up to 34 people had died in the Gekadu region in Guinea, in the forested area, of a hemorrhagic fever, unknown origin. Most people thought it was Lassa fever that causes a less severe disease and is quite prevalent in West Africa. But Médecins Sans Frontières, they had a research base in the hospital in Gekadu, and they decided, with the Guinean government support, to take blood samples and submit those to diagnostic laboratories in Europe. And within 24 hours of those samples arriving, they were shown to be Zaire Ebola positive. That information then went straight back to the World Health Organization, who asked the European Mobile Lab to get a diagnostic unit up into Gekadu in the middle of a forested region, 600 kilometers from the capital, to support MSF's first treatment center. So you can see by the dates that, you know, within a couple of weeks of samples coming in, we had an active, 11 days basically, we had an active PCR diagnostic laboratory. It is the bottom right-hand panel, where the samples will come in from the treatment center. You can see through the gap here, dipterin chloros, extracted and activated in this little CL4 unit here, and then prepared for Altona PCR. And this had a pretty impressive effect on who we submitted to the treatment center and who we released, etc. So this is a little bit complicated, I apologize, but it leads on from work that Ken and Dave both mentioned, the power of sequencing. So each of these little spots, green spots for Guinea, red for Liberia, blue for Sierra Leone, represents the blood sample of an Ebola virus infected individual. 80% of these people died because we were looking at blood samples with high virus load. We did a full genome sequence analysis on what we called a Illumina platform with collaborators, Julian Hiscox at Liverpool University, as he's obviously bringing the samples out of Guinea. And the sequencing analysis with work from collaborators at Edinburgh and Bristol University showed us the relationship between the different isolates, and it showed us over time, because we knew when the samples were taken, and it showed us over location, because we knew where they came from. And it suggested pretty strongly that the virus, initially at the top here, which is in the Gekadu region, spilt over here to Sierra Leone in blue, and then from Sierra Leone and Liberia, it was exported back into Guinea. So an incredible amount of information that was obtained. And when I presented this to WHO and the Guinean government in Conakry back in March 2015, they could see the value of molecular epidemiology, but they didn't want to wait several months for the data. They wanted it to be done in real time, i.e. bring equipment to West Africa and do the sequencing there. And fortuitously, there is a brilliant invention based on nanopore technology, where the DNA slips through the the pores in a membrane. This is the size of the sequencer in front of the laptop there, and information is fed into the laptop, and you get a sequence. So this is amazingly amazing data, some equipment. I can't speak more highly of it. And we then set this up as in Guinea with the help of great collaborators at Bristol University, and showed that the virus was indeed moving around the country. And this helped identify those outbreak locations. It helped shut down transmission chains, and really did support both Sierra Leone, Guinea, and Liberia in their fight against the outbreak. I think one of the best examples of its use was in a flare-up that we had after the Gekadu region, forested region of Guinea, was known to be Ebola-free. In March 2016, there was a case of a young lady that had died in the community. A clever healthcare worker took a swab, put that swab on a diagnostic device, showed it was positive for Ebola. Sample was sent down to Conakry, where the EM lab sequenced it. And really surprisingly, the sequence was shown to be linked to a survivor that we had diagnosed from 500 days previously. So this was a very strange result. The Guinean authority epidemiologist tracked down that survivor. He obviously denied any knowledge of knowing this lady, but he did agree to give a blood sample and a semen sample. His semen was shown to be positive for Ebola virus, and the sequence was near identical to the survivors sequence as well. So, you know, a really interesting event that we're able to link that survivor, male survivor, with the outbreak that occurred. And then this started a WHO program of mass vaccination around survivors to try and prevent this happening again. We've also been looking deep into the forest regions, sequencing samples from shrews especially, and finding completely novel viruses. Another aspect was there filovirus spillover events from bats or other forest animals ongoing before the 2013-2016 outbreak? So with a Guinean scientist who we registered for a PhD in the UK, set up a project of seroepidemiology, going deep into the forest to find examples of antibody responses in bushmeat hunters, deep into the forest, away from any previous reports of the Ebola virus outbreak. So bushmeat hunters are quite an elite group of people in the forested region. Obviously, they can bring back pretty rich winnings from the forest. And they were great in that they allowed us to take a blood sample. And in a lot of those individuals, we found positive antibody responses to Ebola virus and some historical descriptions of hemorrhagic fevers in the villages. And again, these were not involved in the recent Ebola outbreak. And so it brings us back to an outbreak again that was declared in Guinea that some of you may have be aware of. It's in the similar region to the original outbreak, and we all naturally thought it was going to be another spillover event. There's a bit of delay in getting the sequence done. We got support from Oxford Nanopore, who supplied the latest Mania mobile sequencing kit with our colleagues at the Bird Knock Institute in Hamburg. Set up a sequencing capability in Conakry and sequenced the samples of the fatalities that had been identified. And again, the phylogenetics, which is such a powerful entity, showed us that this new outbreak here is closely related to the previous outbreak. We think to that sexual transmission flare-up. So previously it was 500 days that the virus had persisted in a survivor. Now it looks like it's 5 years that the virus is persisting in survivors. So before, when a relatively small outbreaks occurred, human to human flare-ups were very rare, I believe. It was mainly from bats and other forest animals. But now, because you have such a large community of survivors, the chances of a survivor infecting a naive individual are much greater. So I just want to finish on something that Ken mentioned at the end of his talk, and that is that we don't wear a blindfold in emerging pathogens. WHO is very active in surveillance and bringing data together. And I'm part of a panel of international scientists that work with WHO and come together usually on an annual basis to look at all the evidence that we have on those pathogens and what threats they pose to global health. So this is where we exchange the latest intel on small outbreaks that have occurred, seroepidemiology, new pathogens that have been identified. And the last meeting was in 2018. This was the priority list that we came up with. And you can see there that MERS-CoV is on there. And so is SARS, now called SARS-1, because obviously we've got SARS-2. And I remember the conclusion of that meeting in Geneva was that the likely culprit for the next big outbreak was going to be a respiratory coronavirus. And that's why you all are, but we don't probably know what it is yet. And that's why you always have disease X to cover the unknowns. So we also, since 2015, really had a major investment in emerging diseases in the UK. I think we had an underinvestment before that time. And the government under David Cameron established something called the UK Vaccines Network. And Chris Whitty is the chair of this network. And there's 4 working groups within this, and I chaired the pathogen prioritization subgroup. And we came up with a similar priority list to the WHO, similar to the US government, obviously, and similar to that of CEPI that Ken's already introduced. And so every government will have a slightly different view of what they think are the biggest threats, because there'll be different territories that they're involved with and different trade systems that they're involved with as well. But they're all very similar, like I said. And the ones in the middle with CEPI, I was already working on MERS. The UK Vaccine Network was already funding MERS, and that was why the Oxford group could take the adeno platform that they were using for MERS and divert it to look at SARS-CoV-2 back in January, when those sequences were first made available. So I'll touch on quickly about immunity. By far the most cost-effective way to reduce disease burden is vaccination, and it can't be better illustrated by Edward Jenner's discovery of cowpox protecting against smallpox. So this is a Leicestershire outbreak of smallpox, I think, in about 1904. The boy on the right obviously is immune protected, with his classmate infected on the left. Obviously, we would never do this now, but this illustrates the power of immunity in protection. And we can maybe look back at how quickly the vaccine was developed for Ebola virus. There was a prototype virus sitting there, so that initial development didn't need to be performed. This is in 2014. Some phase 1s were quickly trialed. The WHO led the vaccine phase 2, phase 3 studies in Guinea, and obviously showed that the vaccine was efficacious against infection. And then this is Ana Maria Restrepo, the Colombian medic who led the WHO campaign. And we then got finally, the virus was used in anger back in 2015, but it wasn't until 4 years later that it received its full licensure by the EMA and the FDA. But put that into context with the speed of COVID-19 vaccine developments with Oxford, AZ and Pfizer, Moderna, et cetera, it shows that vaccine development can really be accelerated from the usual 10 years to the current 1 year. And CEPI is doing a fantastic job in pulling these vaccine programs together for human disease, but I'm wondering, is there a parallel program for animal disease as well? So in summary, viruses are emerging continuously. Most are RNA and zoonotic. We'll see an increase in incidence as the habits of humans change continuously. We know that there are zoonotic pathogens that cause significant disease in humans already in the UK, and we need to look harder to identify those. I think there is a big vacuum of knowledge around virus movements internationally. People seem to want to be happy to invest in an experimental vaccine, but not knowing what the risks are, where that pathogen is around the world. And in summary, WHO do a fantastic job at bringing that international community together, sharing expertise and information in real time to reduce the impact of global health threats. And early diagnostics is vital. And then it's the last one there, you can never be too prepared for an outbreak. And with that, I'd like to thank you very much.
Thank you so much, Miles, for a really comprehensive and again challenging talk on the the threats that a range of particularly viruses present to us, and I think that sets us up really well for our discussion. And I can see the panel are now joining us. So the speakers are staying with us, and we're also being joined by Maria Jennings, who's Director of the Food Safety Agency. So you're very welcome, Maria. And Perpetua McNamee, who's the Deputy Chief Veterinary Officer for Northern Ireland and is based in DAERA. So thank you all for joining us. And I'm going to try and do the complex procedure of seeing what questions are coming in, which Alistair Douglas is monitoring for me. And then we'll bring those to you as a panel. I'll try and indicate perhaps who should start with each question, but please, all members of the panel, feel free to make a contribution. And I'm going to start with a question that's been troubling me for some time and has been highlighted, I think, by actually all three talks. One of the challenges of SARS-CoV-2 infection, COVID-19, is that we, I think, now understand that a large number of people who are infected are asymptomatic, and yet there's a group of people, and trying to kind of bypass some of the phenotype issues that make it a fatal condition. But my question is there anything we can learn from some of these other viruses or animal viral infection that would help us understand this better? Because I find it quite surprising, the range of severity of COVID-19, from virtually no symptoms at all in those asymptomatic minor symptoms, but then in specific groups, severe infection. Are there any parallels with other viral conditions or in the animal viral infection world, for example, in avian flus, that might help us understand this? Some of the narrative has been around this is all to do with viral density causing infection, but I suspect there's more to it than that, and I'm interested in any comments. Miles, maybe you'll kick off with that.
Sure. So we've done a tremendous amount of work developing animal models for SARS-CoV-2, so we can test vaccines and therapeutics, and some of those vaccines are now licensed. But all our animals are fit, healthy, sort of teenagers, early 20s equivalent to humans. And so we don't see severe SARS-CoV-2 infections because we don't have animals with comorbidities. And so you'll always, I think, see extremes of symptomology in people with comorbidities. And this idea about the amount of virus going in, we can put in 10 to the 100 PFU or 1 million PFU, and we get a very quite a similar outcome. I think in pathology terms, in non-human primates, we definitely see lung pathology that's similar to humans, but nothing like the severe incidents that you get in those thankfully rare cases, because 90% of us were going to be absolutely fine. These are pretty rare cases. For Ebola filoviruses in bats, usually they don't really see the symptoms, but sometimes perhaps they do, but we just don't have enough knowledge. So it's difficult because we, in animal modeling, are dealing with fit and healthy, perfect animals. But maybe Ken might have some more knowledge about the sort of natural environment for the avian species.
Thank you.
Yeah, I mean, we have a project ongoing at the moment. So as I mentioned, bovine respiratory disease is one of our big interests. And we have a project ongoing at the minute looking at genetic susceptibility and resilience to bovine respiratory disease. So what we actually did was we infected a large number of cattle with BRSV and with bovine herpes virus, and we also determined the genetic sequence of all the individual animals. And so we're now at the analysis stage where we're looking to see the variations in disease severity and if we can find genetic markers for susceptibility and resilience. So genetics must play a factor in it, but yeah. we're still analysing.
And will that programme, is that going to allow for the microbiological density of infection as well as the host response? Because I think that's been a kind of interesting narrative around coronavirus, but I'm sure it applies in bacterial, viral and fungal infections.
Oh yeah, absolutely. We're looking at the host response, and we're also looking at how the infection changes the microbiome. So that's another separate project that's ongoing at the moment, where we infect the animals with the virus, and then we see how the microbiome and the respiratory tract changes, and how the interplay between the two can lead to more severe or less severe disease.
Okay. Any other comments on that question? Okay, we'll move on to some of the questions now that are coming through, and one of those is around, I think, the kind of future threats and what might drive those future threats, and I'm interested, I think, to understand if there's some views on migration. A number of you, Miles in his talk, and in fact I think all three talks, we've talked about migrating animals, but coronavirus infection was clearly initially an animal to human spread, but then was spread by human movement. And one of the big mitigations and interventions we've made is to stop people traveling, which is probably being reasonably effective. I guess Australia and New Zealand being very good examples of where reducing human migration has resulted in a reduction in prevalence and incidence in both of those countries. How do we kind of think about that as a transmission issue, perhaps not just for viral infections, but for other infections also? I know, David, would you like to comment on that, maybe first of all?
Yeah, I suppose I would add to migration sort of the movement of food products as well.
Yeah,
moving around the world in a much more free and rapid way. You know, yeah, a lot of the zoonotic agents that we discussed are very much related to animal products of animal origin, but increasingly we're seeing natural produce being very much linked. You know, STEC is very much linked with natural produce, and where we have these infectious agents remaining viable because of the very rapid movements around the world and the very controlled environments, then we can see a different level of exposure and a change in the epidemiology in the human population as well. What would help to mitigate that? Well, you know, ensuring that we have control measures where these things are being produced, and then of course being able to identify them before they come into a particular jurisdiction. And I mentioned the metagenomics, you know, where we might have a novel pathogen, for example, that German outbreak in 2011. You know, that type of STEC had not been seen before and linked with disease, so it had quite a novel combination of variance properties. And the genomics age allows us to identify that rapidly and actually then try to, actually it was quite difficult to find the source of that because it was bean sprouts, which nobody remembered having eaten in the meals which were implicated, which is very much the case. So I would add the rapid movement of foods around the world as well as populations of people and animals.
Well, thank you. And maybe Perpetua or Maria might want to comment. Yes,
yeah.
Thank you.
Thank you very much, Stuart. And first of all, I'd just like to say I really enjoyed those presentations. So thanks to David, Ken, and Miles. And just to pick up on David's point, the movement of food globally and internationally is increasing all the time. And one of the good examples, I think, over the last couple of years is the work that we do in the ports in terms of surveillance, where we picked up some food product, some pork product, which then AFBI analysed and found some fragments of swine fever. So it was a really good example of collaborative working. We were able to intercept a potential pathogen that could have been catastrophic for our pig industry. We were then able to raise awareness through the pig industry using social media platforms but we were also able to directly focus on groups of students who were coming in from that area in Asia, you know, to warn them about the risks associated with importing food products. So it's a really nice example of you know, the work that we can do collaboratively to protect our agri-food industry.
Oh, thank you. And Maria, would you have any comment from a food safety perspective?
I would indeed, Stuart. Thank you. Also, I echo Perpetua's thanks to the presenters. The presentations were fabulous. Just to say that we have been looking at in increasing and improving our surveillance techniques and looking at data sources across the world to try to track how you know, food problems with food or issues related to food track across and move into the UK, and that obviously is a lot easier because of the technology that we have available to us now that we didn't have even 5 years ago. But actually that only picks up the known problems, so the things that individual countries have already identified and that may have a knock-on effect for the UK. But David talked earlier about disruptive technologies around the work that AFBI is doing, and those techniques actually now link issues. So where there may be individual food poisoning outbreaks or incidents of some kind, we now through the science can link those incidents up together to show that possibly, you know, there was a linkage between different stages of how the, you know, either animals or food had been affected by a particular pathogen. So you know, we have at our fingertips now much more capability than we ever had to try to deal with these big issues across the world, not just, you know, within Europe and within the UK.
No, thank you. And that very nicely takes us into another question, maybe for the panel, around the power of sequencing. Miles showed us some lovely data on how powerful, particularly rapid sequencing can be in understanding outbreaks and pandemics, and allows then a much more focused and I think evidence-based intervention to try and mitigate whatever's going on. And maybe but back to you, Miles, that you showed us the Oxford Nanopore, which we're using in Northern Ireland as part of the COG program to do some rapid sequencing, and it has been truly just amazing how such a small device can give you pretty good data in a rapid turnaround. We hosted the Prime Minister last week on his visit to Northern Ireland, and he was shown the nanopore technology and was really quite blown away by just the power that we can now bring to the sequencing world with technologies that 5-10 years ago we absolutely couldn't have dreamed of. Where do you see that going, Miles, in terms of sort of much more routine use and really baking that into our future surveillance programs?
Yeah, I think, I mean, the old days, PCR was a fantastic invention, and it was a disruptive technology, and allowed some incredible insights of pathogen identification and speed. But now, I mean, I always take a MinION around with me in my pocket everywhere in the world that I go, because you never know when you're going to need one. But I just remember being in Conakry in an old hospital room that we set up a lab. I showed you the picture of with this young PhD student who just came in with some MinION and a laptop and some kit. And after getting from the airport, by that evening, all the things were prepped, everything, and we got the first sequence of Ebola virus within like 11 hours, which was pretty amazing. And the information we had then, we just were shocked where we knew it came from. It didn't come from, well, it came from Sierra Leone then, from one route in, and another route in, it was coming in from the forestry region again. So that information, you can give to the authorities, and they can rapidly shut down these transmission routes. So I think mass PCR testing will be in the future replaced by mass sequencing, but we need to get the bioinformatics end to catch up with the sequencing hardware. We need to train a whole new cadre of bioinformaticians, though that is being done. And we need all the pipelines that can rapidly process the data. Computing power is also increasing. So I think, we will get there, but we won't be doing PCRs, but we'll be doing MinION sequencing or other platforms. I haven't got shares in them. I wish I had, but it's fantastic technology.
Maybe I could add to that as well.
Ken?
Not just the sequencing, but where we get the samples from. So we're seeing developments in technology such as aerosol samplers, which sample the air for aerosols, and these can be set up in like high traffic hubs like international airports or doctor surgeries, and these constantly sample the aerosols that are there. And so you can then take that and sample and see which viruses are in circulation. And then with COVID, we've seen how wastewater sampling has come into its own as well. So there's different areas that traditionally we wouldn't be thinking of, but where we get the samples from for doing the next generation sequencing. I think we're going to see a lot more of that in the future also.
No, absolutely. I think. Go ahead.
Ken's point. I think sampling is fantastically important, and where public health is going is especially the sampling of wastewater, sewage water. The amount of information in there is incredible, and there's some great visionary people in the Joint Biosecurity Centre who are going to be part of our new agency going forward, with some great vision of how sampling, especially sewage water, will give us those early surveillance systems.
And just a kind of follow up to that, do we see that as something that might be applied not just to human health but to animal surveillance, because we can get at the excrement from animals in particularly large agricultural situations that might provide a very real-time surveillance opportunity to make sure we're monitoring animal health and also those potential crossover situations?
I can probably jump in a bit, just from my previous experience, and I'll take you back 20 years ago. This work has been going on for many years. I spent some time, 2 years, in the Central Pacific region, where animals and humans lived very, very closely together. The population density was very, very high, and the wells were co-located basically with the pig pens. I worked with WHO in monitoring some of the wastewater and the excrement coming from those animals, and it was very clear, and we proved that there was a correlation between the zoonotic pathogens coming from the animals and the infant mortality and survival rates at 12 months. We did a lot of work at that time with the WHO, and over the last 20 years, we have monitored the incidence of infant mortality over 20 years, and we can see that by education, by raising awareness, by public health programs, we have managed to increase the infant survival rates. So that's a very sort of simple example. It's a very long-term outcome in terms of the links between surveillance, testing, working between animal health and human health, and long-term outcomes. And I just wanted to share that with you because, this has been going on for many, many years in the animal health field, as well as in, and linked together with public health.
No, thank you.
So I hope that's useful.
Very useful, and I think these new molecular technologies allow us to do it at a degree of precision and at a scale that would have been very difficult using more conventional microbiology in terms of culture technologies, or even with PCR, which is not entirely straightforward in wastewater because of inhibitors. I'll just share a very dystopic conversation that we had at a SAGE meeting, which was trying to understand whether SARS-CoV-2 was an excrement, and then there was a conversation around might we aerosolize this when we pass wind, and it kind of brought a whole new kind of dystopic kind of thought around the current pandemic, but it actually it illustrates that we've got to think differently. We've got to think about whole ecosystems, and I think those comments around sampling and using these technologies was really helpful. We're coming towards the end, and there's a further question which would be interesting to hear from each of the panel around, which is what do you really see as the future threats in animal and human health? I'm kind of interested in viruses, bacteria, and fungi. I kind of think fungi are one of those under-investigated parts of the ecosystem that clearly climate change is affecting, and we're seeing particularly, for example, for Aspergillus species, antifungal resistance developing, and that's spreading in ways that might be explained by the changes in the current climate systems within the world. But I'd be kind of interested to go around everyone, and we'll maybe finish with you, Miles, because you've perhaps the most global view of this. But maybe starting with Perpetua and Maria, what in your worlds, what concerns you most? Maybe start with Perpetua.
I suppose what concerns me most at the minute, Stuart, would be climate change and global warming, and how that can impact on the spread of tick-borne infections, further up in a more northerly direction. And I think that would be my biggest concern: tick-borne zoonotic conditions and tick-borne animal health.
Okay.
Non-zoonotic conditions also.
Thank you. Maria, anything from the food safety and the future? Where are your concerns? I lived through the E. coli 0157 situation. We actually had a conference in Hamburg at the during that outbreak, and half the conference didn't come, particularly people from Spain, because they were being blamed for it at the time. But but it turned out to have nothing to do with Spain, and I think it was cucumbers. But where do you see the kind of future threats around, particularly infection?
That's right. I don't think Spanish cucumbers have recovered ever since. The levels of foodborne disease have remained stubbornly high for us in the FSA. We monitor, obviously everything at a national level, and it has been really difficult to drop those levels down. The big, emerging threats for us are antimicrobial resistant bacteria, and that's a real concern, because if you don't have the methodologies to be able to deal with the actual infections themselves, then there are going to be more longer term implications for people who have to live with illnesses, and also who ultimately may die from those infections. But also, we're very worried about intolerance as well. So food intolerance and allergic reactions are really high up on our agenda, and we're doing quite interesting work around that. And there's there was some talk earlier about the microbiome, and I think that's really important in terms of looking at allergies and intolerance. And then on the viral side, obviously norovirus causes a lot of illness, so human illness. So again, big numbers in there, and may sometimes be foodborne. So that's definitely a concern for the Food Standards Agency.
Well, thank you, and hopefully sequencing will help at least with the antimicrobial resistance to at least identify if genes are present that would be of concern. David, in the world of bacteriology, what are your what keeps you up at night in terms of future threats?
Well, I mentioned a number of them in the talk. Campylobacter causes the biggest number of cases, but the severity is not as high as it would be with something like Listeria, which is very many magnitudes less in terms of overall incidence, but a mortality rate of over 17% seen in 2019 in Europe, and a hospitalization rate of above 80%. So that's very much focused towards the immunocompromised populations, are becoming, of course, a greater percentage of the total population. So, food supply chains need to be able to protect all citizens. It's something I've actually grappled with myself and changed my view over time. Should we expect the food supply chain to be pathogen-free? I think ready-to-eat foods absolutely should, but it's only as good as the pathogens we know. If we talk about raw foods, we've mentioned the microbiome. There are benefits in us being exposed to a wide range of bacteria, and the benefits from that. So I, for one, would not want to be eating a sterile food. I want to be exposed to the diversity of the microflora within that. But there's a balance there, and we have to make sure we protect people. Ultimately, AMR is the thing which causes me the biggest amount of concern. It truly is a global challenge. The one good thing about AMR, if there is that we know what it is, and we've got tools to detect it and hopefully begin to understand what the drivers are. But I don't think all the drivers are fully known. I've got a range of strategies that we are applying to that. But we need to make sure we have absolutely concerted effort, because one weak link in the whole ecosystem supply chain could wreak havoc really with the outcomes we want to achieve.
No, thank you. And yes, how we deal with low and middle income countries and how drugs are used both in human and agricultural contexts, I think is one of those important drivers we need to understand better. Ken, from a more virology perhaps perspective, and clearly influenza and the interactions between animals and humans is really important, but is that what keeps you up at night, or are there other areas that you think are going to...
Flu, I mean, another Spanish influenza obviously would be devastating, and probably the biggest candidate for that is that these H5 viruses do work out how to start transmitting between humans. I mean, we've had them for over 20 years now, and so far we've been lucky it hasn't happened. With flu, it's probably not the virus that you're looking at, but it's one that you haven't noticed before that takes you off guard. So, and we know that the pandemics happen pretty much once in a generation. They average once every 25 years there's a pandemic. So it's not a case of if it will happen, it's when it will happen. But we're in a better position now. I mean, we have flu vaccines, although the pandemic vaccines are maybe, they don't often behave as well as what they should. So, I mean, there's definitely work to do there. We've seen the 3 SARS, or the 3 human coronaviruses since the turn of the millennium. Could there be another one? Possibly. I mean, from an animal perspective, probably the one thing that keeps me awake at night is African swine fever. It's moving west. And the worst case scenario would be that it got into Northern Ireland. But thankfully, we're an island off an island, and hopefully that won't happen. But yeah, that also keeps me awake at night.
Thank you. And Miles, can we leave the final thoughts to you in terms of what keeps you up at night in terms of future infection threats in both animal and human health? Yeah,
I mean, your other guests have already highlighted the key ones. And I mean, AMR was on the risk register, the government risk register, high up there before this outbreak, and it needs to be back up there. Well, it's still there, but we need to ensure that we fund properly the work to reduce the incidence, and also supporting the pharmaceutical industry in developing newer, better drugs as well, because that is a major gap in our medicines cabinet at the moment. So you're thinking about the worst pathogen you could have, and this is what we think about in the WHO sessions on pathogen prioritization. And so you think of something that's really highly transmissible, no vaccines or therapeutics, and has a high case fatality rate. And Ken already mentioned another Spanish flu. That's a good description of one. But another coronavirus, which would be just as likely, or another virus we haven't even heard of yet. One of these great unknowns. But we can't prepare a vaccine for everything. But what we can prepare are platforms, our diagnostic platforms that you've quickly got into action with your SARS-CoV-2 diagnostics. We need quick ways to check the medicines that we've already got licensed. Would they have effect, which obviously has some benefit against COVID-19? And we need these rapid development technology platforms, like we've got mRNA now that's been into hundreds of millions of people, and that's, I think, a massive piece of progress for future pandemics as well.
Fantastic. Well, thank you very much. I'd like to just finish off by really thanking the panel for a terrific session. I really enjoyed it, and I think we could continue on having more discussions on some of the really important areas that have been raised. So thank you to all of you for the fantastic contributions. Thank you for the questions, also from those who have put those up in the chat. And I'd like to just use this as an opportunity to thank the team at AFBI, Ken, Alistair and team for just the superb response that we saw during the COVID pandemic in terms of delivering really critical PCR testing. I think we'll need help again over the next number of months, but I think into the future there's a really important conversation around the diagnostics for disease across animals and humans, and indeed sequencing pathogens in plants is the same technology. So I think there's an opportunity in various parts of the world that we think inclusively and cross-sectorally around how we're making sure we've got the right sampling, the right diagnostics, and then the right interventions from public health in animal contexts and public health in human contexts. So thank you for all that AFBI has done, and thank you for the support from the Minister around all of that. So it's my pleasure now to hand back to Elizabeth, who's going to, I think, just close this session and allow us all to go for some lunch.
Indeed. Thank you, Stuart. So, folks, if you didn't know before, you certainly know now, and this session was not designed to scare the life out of everybody, but I think we have definitely raised awareness around the bacterial, the microbiological side of things and the virology side of things, just the amount of threats that are out there. But I equally hope that you are assured through all the talks, and as David's talk, Ken's and Miles, that there's very strong diagnostics platforms, surveillance platforms, vaccine development platforms, interventions being trialled there. And whilst there's many threats across the world and on our doorstep, there are systems in place to protect us from those. So I just would like to say thank you so much to Stuart for chairing the session so well. I certainly found it absolutely fascinating, and thank you for your time today, Stuart. Thank you to Miles for giving his time. It was fantastic to hear from Miles, such an expert in this field and working at the heart of government. Thank you to Ken and David as well from AFBI. Thank you to Alistair Douglas behind the scenes, who's been very much working to pull this session together. Thank you to Perpetua and Maria, who have very much fed into the panel discussion very expertly with their important knowledge. So thank you, ladies, for your time this morning. In recent days, there's been announcements from DAERA that they were looking at options for a new vet school, and I think this is very much part of those discussions, and I know in AFBI we're very excited about that possibility of a vet school coming to Northern Ireland with Queen's and UU, and AFBI can very much play a role in that as well, linked with the expertise that we've heard from today. So folks, it is time for lunch. Please go and stretch your legs to get some lunch. If I was at home, I'd be feeding pet lambs, so you've saved me from that today. But do join me back here at 1 o'clock when we'll go into our next scientific session, which will focus on modelling and managing our ecosystems to drive future sustainability. And as I say, that's very much aligned with our agenda on enhancing the natural and marine environments. So thank you for your time this morning. Grab some lunch and we'll see you back here at 1 o'clock. Thank you. Bye-bye.